A very good day to all of you. My name is Koert van der Speten. I'm a surgical oncologist and pharmacologist from Belgium. Is a scientist, a renowned scientist from the Netherlands, who is currently the professor of surgical and translational tumor biology at the University Medical Center in Utrecht. Might sound like a little bit strange to you that scientists are going to talk today in a community of mostly clinicians. One have is to show you that these two. Before we embark on the conversation itself, I want like two opening statements. I'd be a little bit provocative. The first one is that the current TNM classification, as we use as surgeons and as clinicians, has come to its limits. By this, I mean that TNM, which is on radiological findings, that we have all metastases. And we are clinical trial. I'm looking at all lung or liver or peritoneal. We tried the same strategy in all of these different metastatic sites. And one of the things we're going to talk about later is that this might not be a very wise idea. Understanding a bit of the tumor biology, as we will later, we will see that these are very different animals, and probably there is cause to think, or reason and rationale to think, that a different approach, more based on the site, is more appropriate. So that's the first one. The second one is another provocative one that, and I keep telling it at different places where I talk, is that the best thing that has happened to this community of surgeons and clinicians and scientists interested in peritoneal surface malignancy is that we had a negative trial, namely PRODIGE 7, which kind of like put everybody in a search. This has led both to an increase, a search really, in both new clinical trials, better strategies in doing these trials, but also in a lot of translational research. And what is really, and this is the reason why I'm admiring Ono so much, is that the world of clinical surgeons and medical oncologists doing clinical trials and the translational scientists in the lab, it's no longer two islands. We're going to see some very specific examples from Ono's lab that these worlds have come like really close together, and that results of his research have a direct impact on the clinical trials we are trying to construct these days, and how we take them into the clinic, and hopefully with better results for our patients, because that, of course, is like the goal. Now, with that being said, we're going to talk about organoids today, organoids as a model of researching different metastatic sites. And it might not be clear to all of our viewers and listeners, but Ono, maybe you can tell us, what are organoids? Why are they so interesting?
Thanks for the question, Koert, and I couldn't agree more with you with everything that you said so far. So organoids are a novel culturing system. I'm saying novel, but actually it exists already for about 10 years, I think, that we saw the first publications on organoid technology. Before, we were using cell lines. And I think the major difference between organoids and cell lines is that organoids are really representing the tissue of origin. They maintain the genetic and the phenotypic characteristics of the tissue of origin or the tumor of origin. From a genetic perspective, you see maintenance of, for instance, driver mutations. You see also histopathological maintenance of phenotype. For instance, if you derive an organoid from a mucinous tumor and you put it in a mouse, you see back this mucinous histology. And really, it's based on novel insights in stem cell biology. Tumors are driven by stem cells, as normal tissues are, the regeneration at least. So insights into that stem cell biology, what makes a stem cell renew itself? What makes it differentiate? What makes it generate mass? So that insight was used to recreate in a dish, basically. So to add components of that surroundings to the culturing medium and to the three-dimensional matrix that they see in a tissue, also to recreate that. And in that sense, once you take tumor cells, for instance, from a colon tumor or a peritoneal metastasis, and you would put that in the original medium that we would use and on 2D in plastic, then these tumor cells, they experience something that they've never seen before. And most of them cannot handle it. So you get an enormous crisis. You get an enormous selection for tumor cells that are actually adapting to this old-fashioned, let's say, traditional culturing conditions. With organoid technology, we take away that crisis. We try and mimic the conditions that the tumor cells were used to when they were in the body. And therefore, you get this maintenance of genetic drivers, but also genetic heterogeneity, for instance. So the multiple subclones that can exist in a tumor are maintained in the organoid culture. And that creates a huge platform for all sorts of research. I think as an introduction to organoid technology, that would be sufficient for now.
Yeah, one of the things that took me some time to realize when this technology came, let's say, on the market, was that, and you have to correct me there if I'm wrong, you basically are creating, like, with having different organoids from different patients, you're creating like your own living library of patient cancers, where our previous models based on cell lines was mostly on immortalized cell lines or anything, but was less representative of what we are facing in the clinic. Is that correct?
That is absolutely correct, and I think that is the way forward. That is one of the major strengths of the technology. We used to use 2 or 3 cell lines and made the old-fashioned way, and that would then represent colorectal cancer. But the disease is so heterogeneous, and in the introduction you already alluded to differences in, liver versus lung versus peritoneal metastases. These are different entities. So the technology allows us now to create these living biobanks, as we call them, in which the heterogeneity of that specific disease entity is fully represented, or at least represented as good as we can. And then I'm talking about, for instance, genetic diversity, because, for instance, peritoneal metastases with mutations in BRAF may respond or act or grow very differently from those with different driver mutations. So from a genetic perspective, but also from a histopathological perspective, within the peritoneal mets, we recognize the adenocarcinomas, of course. We recognize the mucinous carcinomas, the sigma ring cell carcinomas. It's important that we start creating these biobanks as a platform for studying the disease, and that's what I think at this moment is the one of the major strengths of the technology.
What I liked really very much about the studies that we're going to discuss here is the fact that you categorized your organoids, or your organoid library, at least for this part of your research. You categorized them based on the metastatic site in the patient that they are representing. So you have organoids from patients who developed lung metastases, you have patients who have liver metastases, and you started like a complete, like really intriguing research trajectory on that, both looking at why these patients are specifically, so based on the organoids, looking at why these patients, one patient is developing lung metastases and the other one is developing predominantly liver metastases. And then you took it all the way to potential targets for treatment. So that is like a huge trajectory. And when I was reading this overview article, I was like intrigued, like, how long did this take you from concept and having the organoids to the point that you're telling us, well, this population is like that, and we have a target there, which can be targeted either by systemic therapy or by other means. What is the timeline on such a discussion?
Yeah, this took a long time indeed.
But you could you, because I think there might be a little bit of a misunderstanding here. What is long in your perception at this point? Is this now like 5 years, 10 years?
Well, the study itself started with the organoids being present, but that's what I meant with the strength of these living biobanks. That once you have a research question like this, in this case, my lab has a long-standing interest in mechanisms underlying metastasis to the liver, to the lungs, to the peritoneum. But this living biobank now allows you to select organoids that have a certain phenotypic difference in order to be able to compare them. Mm-hmm. Right. So if you ask me how long did it take you to do this study, then first of all, we needed to make the biobank. And that, of course, took us a number of years. That was one project. And in that project, the goal was to generate organoids from, in that case, microsatellite unstable metastatic cancers, which are not very common, as you know. They're only 3 to 5% of all metastatic colorectal cancer. And so that took us some years, but then we were able, seeing those different metastasis patterns in the clinical, or at least in the patients that were included in this study. Then we thought, okay, let's see whether that is now preserved if we transplant these organoids in the mouse colon. Okay. So this is what we did, and we had one metastasis that was derived from, one organoid that was derived from a lymph node metastasis, and in this patient later on also developed lung metastasis. One was developed from a liver metastasis, and the other one was anonymous, so we don't know the clinical trajectory. And it turned out that indeed a lymph node-derived metastasis went to the lungs in the mice, not to the liver. The liver metastasis-derived organoid went to the liver, but not to the lungs. And the one from which we do not know the clinical history went to both organs.
It's like very, very intriguing, because in our conventional surgical thinking, we have this idea that you have your colonic cancer, and basically tumor cells evade, will follow the portal vein, will go to the liver, introduce their liver metastases, and then as a kind of sequential event, it will spread from there to the rest of the body, and this might be the lung. So basically your findings there from the lab are directly contradicting that these are not sequential events. These are just like totally different animals. With a totally different kind of evolutionary thing there happening. So that on itself has already like very big consequences to treatment, isn't it?
Yes, but it also tells you that, it reinforces again and again that colon cancer is such a heterogeneous disease. Because, of course, from the clinic, we also know patients that have lung-only metastases. So in general, the dogmatic view of metastasis is indeed, as you described, you get liver metastasis, and then from there metastasizes onwards to lungs and other sites. But there is also clinical evidence that lung metastasis may in fact be, or can at least, originate from a different pathway, not needing liver metastases, because these patients are just there. Maybe not the majority, but they are there. Mm-hmm. And I think this is how you also need to see our study. This is not the model for lung metastases, because I'm 100% sure that there will be patients whose lung metastases derived from the liver metastases. We just don't see that in this particular model representing this particular patient.
Correct.
And if you look at genetic studies where they looked at, and I'm referring now to a paper by Kamila Naxarova, who we also did who we did a study with, this particular study that we're now discussing. In 2017, she published a paper in Science showing that 65% of the liver metastases were not originating from lymph node metastases. So that's the vast majority, right? So this is just our dogmatic view of liver metastasis formation. We have a colon tumor, you have tumor cells traveling through the vasculature, and the first organ that they see is liver, and then they grow out. But the surprising thing is, of course, the other 35%, which is a minority, and these were related to the lymph node metastases. So why do liver metastases need to go through the lymphatic vessels to be seeded? And so I think how these let's say the topology of how metastases occur, the pathways, the anatomical routing, so to speak, is something that, of course, you cannot study in patients because it's very fast, and you cannot use patients for these kind of complicated questions. But for that, the mouse studies are really complementary. So genetic studies, comparative studies on metastasis sites, which metastases are related to which part of the primary tumor and to each other. That is very insightful to give relationships between metastases. And then the mouse studies can be used to actually model and to ask cause and effect relationships. And that's what we've done in the study that we're discussing right now.
It's one of the intriguing things I took back from reading this manuscript is that once you're told about the timeline, it takes you quite a bit of time to build this organoid library. But once you have it, the timeline of putting a research question into your animal model is actually quite short because it is available for you, and then basically you can take that. And I also just liked your comment about the fact that you select a little bit in your organoid library the two extremes of a spectrum. So one patient who really, or a few organoids with dominant liver metastases, dominant lung, and you try to find and get insights from that and see.
These biobanks are really a resource. So and to be used for many researchers, right? And that's why now we're also creating a catalog, a searchable catalog, so that people can actually search what is there, because other researchers may be interested not at all in lung versus liver, but they may be interested in studying KRAS drivers versus BRAF drivers, or patients that lived for more than 5 years versus those that died within half a year. So if all that is annotated in the catalog, then the organoids are there for the studies that they can then be used for. And this is how we went about selecting the organoids for this particular study. And then just to comment again on the timeline, then it took us about 4 years to do all the experiments that are described in the paper. But that's starting with the organoids that were already present.
Which is a huge amount of work done in for me a very short time. Would you care to elaborate a little bit on the things you saw on the research project itself, in particular to the fact that of course as surgeons we know as a general principle that there is intermetastatic and tumor metastatic heterogeneity, but specifically in this paper you were referring to that if you look to heterogeneity and evolution of liver metastases, that they are totally different from lung metastases. Could you elaborate a little bit on that?
Yes. So there are, I think, two major ways in which you can study this. One is the genetic way, in which, of course, now we know this single-cell sequencing approach, where you can sequence the DNA of every single cell. And then see which cells end up where and which cells give rise to which metastases. So by comparing, for instance, the signals out of the primary tumor to that of the liver and the lung metastases in this case, we opted a slightly more, or let's say a different approach, and this is called optical barcoding. Optical barcoding, so we give every cell a unique color, and we do this by transducing these organoids with lentiviruses encoding fluorescent proteins, 3 different fluorescent proteins. And then depending on the number of viruses that these cells take up and are integrated in their genome, and depending on the location of where these viruses differ the colors. And those finally determine the final color of that particular cell. So you get a sort of confetti type primary tumor, and this you can see in the pictures, that all kinds of colors, starting out with only 3, there's all kinds of colors that we can identify in the primary tumors.
Okay, am I correct to assume that you put a kind of colored timestamp on your tumor cells, your organoid, at the starting point of your research, and then you can follow them both at the site of the primary, but also at the metastatic sites, and see what happens with your colors as a kind of stochastic evolutionary picture of what is happening here.
Exactly, and here there were 2 readouts that were really important to us. One is the primary tumor is extremely heterogeneous, so the color diversity is very large. This is the starting point, right? This is what we created. And now the question is, how color diverse are metastatic sites? And then the surprise came that when we analyzed the liver metastases, they were always, well, not always, the vast majority of liver metastases consisted of a single color, and sometimes 2. And we interpret that as a cell doublet seeding the metastasis. But the fact that all metastases, regardless of the specific color, always consisted of one single color meant to us, or told us, that it's always single cells that seed these metastases. Because if there would be clumps of cells, they would have different colors, and then you would see multicolored metastases.
Correct.
And that's actually what we saw in the lungs. The lung metastases sometimes had a single color, but the vast majority of lung metastases were multicolored. And that was actually the starting point. So that was one important readout. And a second important readout was what we describe in the paper as the evolutionary bottleneck. So how many colors from the primary tumor can actually generate liver or lung metastases? Now, is there a selection against certain colors that do not have the capacity to seed metastases? And then you see that this is really true for the liver metastases. There is a selection, there is a bottleneck. Not all cells can make it from the primary tumor to the liver. You have reduced color diversity if you look at the liver metastases in the entire organ. The colors are missing. And this in the lung metastases we did not see.
Okay.
So color diversity, if you look at all the metastases in the lungs, is similar to the color diversity in the primary tumor. And that said to us that virtually all tumor cells, regardless of their color or their genetic composition, can actually make it to the lungs. But this is not true for liver metastases. Here we have really a bottleneck.
That immediately puts, as a clinician, the idea for me that that the majority of systemic chemotherapy in patients with metastatic colonic cancer is based on a pathology of the primary tumor. Now, if I translate what you're just saying, then especially in the case of patients with isolated liver metastases, I should rather target my therapy based on that one color, as you say, or 2 colors. So we should get a biopsy of the metastatic site, and if we want to have a far more effective systemic chemotherapy in these patients, it might be better to take a biopsy from that and target your therapy to that. Because at the same time, this was like a little bit of a contradiction to me, that despite the fact that liver metastases are, let's say, super selected, do you say, from the primary, we still know that patients with isolated lung metastases, being post-clonal, that they are still doing better.
Yeah.
And maybe part of the answer to that question is in the fact that probably if we do the systemic chemotherapy and liver metastases based on the primary tumor, we might be shooting completely in the wrong direction.
Yeah, there are several aspects to what you're saying that are super interesting. I think we do not know yet. We do not have predictive biomarkers for response to systemic therapy, right? Apart from maybe EGF receptor targeted therapy. So if you look at driver mutations, those are similar all over. So the differences So it doesn't matter if you take a biopsy from the primary or the liver or the lung metastases. So it's something else that is distinguishing these lesions, and that also cannot explain why indeed patients, for instance, with lung-only metastasis do better than patients with liver metastasis or peritoneal mets. But there's one aspect that you in the introduction said or remarked upon, and that's path to metastasis is the path itself, right? It's the routing. How do they get there? What is the mechanism of their initiation? But then once they're there, there's the what happens then? And the lung microenvironment, of course, is completely different from the liver and the peritoneal microenvironment. So lung metastases being polyclonal immediately after they are seeded does not necessarily mean, for instance, that they are more resistant to therapy or that they're more aggressive, because they will have to deal with this highly pro-oxidant environment that they find themselves in. And these lesions often don't grow or grow very slowly. And that there may be secondary processes during the outgrowth that also determine very much on what the metastasis that is finally visible on a CT like. And I think that aspect, that sort of later aspect, we cannot model in the mice simply because most of them also develop peritoneal metastases, and that defines their humane endpoint. So then we have to stop the study.
Mm-hmm. You were most of all of the work in this specific studies on both lung and the liver metastases. Of course, our audience today is peritoneal surface malignancies to a large extent, and the question immediately comes, of course, do you have like similar work going on in peritoneal in patients with, or in organoids derived from patients with peritoneal metastases, peritoneal surface malignancy, and to what extent do they concur or differ with what you've just been saying?
Yes, we've done a very similar study also with Kamila Naxarova, and that study is now finished, but we still have to submit it to a journal, so I can't say very much about it. So we make an appointment again in 1 year, and then we'll do one special on the peritoneal mets. But that's super interesting.
It is interesting indeed, just the fact that, and it's a consequence following on the ideas we were just expressing, that this is once again another entity and which will have its own specificities. New research is going to be a lot of hypothesis generating things there, but the link to the clinic is like quite short. That if you find like either on the genetic level certain mutations or whatever, or facilitating conditions, which you found for lung with some of the facilitating conditions, there might be both targets for systemic therapy and more like targeting, but targeting in a different way than we have come to understand until quite recently. Until quite recently, targeting was like based, we have this mutation and we're going to do this. Now a lot of other things are coming in place there. When you were talking about,
I think the context dependency of so that's not an issue now. The way we treat colon cancer. I completely agreed with you in your introduction when you emphasized on these context dependencies. You know, the lung microenvironment and the peritoneal microenvironment are completely different, and we can even reproduce that in the mice. In one study, we used, for instance, mice, which we also gave a human immune system, so we could study immuno-oncology.
Okay.
And in those mice, liver metastases were completely eradicated by immunotherapy.
Okay, because
so killed by human T cells.
Okay.
But the peritoneal metastases in these same mice, they were completely refractory, and that is something that is also clinically observed. Patients with peritoneal metastases and ascites formation are resistant to in the immunotherapy trials, or at least respond much less than patients without peritoneal mets and without ascites. So that's again to highlight the value of organoid technology, that you can recreate these kind of clinical problems. In this case, in mice with the human immune system.
That's like really interesting, because this was, until quite recently in my thinking, was kind of a kind of blind spot when you were doing organoid-based research in animal models, that the majority of work I've seen so far has been, of course, in immune deficient mice, and knowing both of what is going on in the clinic with all the immunotherapy trials, and also from the more like basic point, translational point, what is the role of the immune system, the microenvironment, these tumors. So it seems that already that is being addressed, and that we are putting an additional layer on the limitations of the organoid technology as conceptively. That is like really interesting.
Yeah, and obviously, I mean, every model has its drawbacks, right? It's not perfect. The perfect model is the patient itself, but we are very limited there in what we can do. Organoids are already that's, let's say, step one, a very good representation of the tumor cells inside the patient. And now, we need to use immunodeficient mice, otherwise they wouldn't grow out at all, because the mouse immune system would kill human tumor cells. And that's the system basically that we used in the paper that we're discussing. Obviously, the absence of an immune system of which we know that it shapes metastatic patterns is a drawback in that system. Nevertheless, it's always about the question that you're addressing, right? And in this case, it was mechanistic differences between liver and lung mets.
Correct.
And apparently, the immune system there did not play a role. That is not to say that if we would reproduce these models in humanized mice, that if we look at immunological aspects of clone selection, if you're talking about methods, that we would find something different. That's possible. We didn't do that. In any case, what we did find, because we did put these same models in humanized mice, and then at least the patterns of metastasis are completely preserved. So in that sense, the presence of an immune system did not make a difference.
Okay, that's like very importantly. You once again pointed out there that what took a very long time for this surgical community to understand is that the patient is not the model where we're going to address these research questions, and that once again was the big good outcome of the negative trial, that we understand that we need to isolate these questions in a controlled environment, generate a hypothesis and a potential treatment strategy from there, and then retest it in the clinic. Oh no, if I hear you, basically organoids are ideal. There is no drawbacks. They are perfect. I can tomorrow I'm going to start doing this in my backyard. Is it as simple as that?
Well, it's like I said, every model does have its drawbacks, but I'd like to focus on what they can offer, the value that they do offer, and I think there is the largest benefit is in representation of the disease in all its heterogeneity. For colon cancer, it's relatively simple. The protocol is relatively simple. Many people are doing it. Many people are building and have built living biobanks using organoid technology. So yes, if you want to start tomorrow, you can, if you have the money. That's one drawback, by the way.
Yeah. Okay.
It's not cheap. Yeah. It's much cheaper to culture cell lines. But if you would find the funds to answer specific questions using organoid technology, then you could start tomorrow. And you can come to us, because we give training also for people who want to learn organoid technology.
Okay.
But then within a few weeks you can start up your own lab and do it.
Okay, it's like interesting, because in the beginning it's almost like an ethical question. I was a little bit concerned about organoid technology, because the media and things you are using at some point, this was going towards patents and things like that. Now I hear also from your earlier comments that we're going a totally different direction. If you say we're talking about cataloging this and have like an open catalog of what is available of organoids, and sharing them and sharing the protocols and the media. This was one of when the technology started, I was like concerned about that this was going to be like an industrialized kind of environment at a very high price, and that the clinical relevance to our patients would be like limited by that. But I'm seemingly I'm quite wrong on that in recent developments.
Well, it's not a crazy thought at all, and that was also our concern when we started to do this in a sort of a standardized fashion, generating organoids in clinical practice. Obviously, we were limited and are limited budget-wise, because you cannot do this for every patient, every sample that you get, but also from an ethical perspective. So this is about 5-6 years ago. We initiated the Utrecht platform for organoid technology, and that's a facility here at the hospital that helps researchers get access to patient-derived tissues, in a way that is in which both the logistical aspects of getting that tissue are optimized, but also it's based on an informed consent procedure, an ethically approved, legally sound, informed consent procedure. So patients are explained what it means to have their organoids, which are in principle living forever even after they are dead. There is no endpoint, is similar to the cell lines, but also in many cases they are sequenced, potentially generating information that could lead back the organoid to the individual. Right. So all of that is recognized and is described in the protocol and is explained to the patient, right? And so now we have a format where our facility, Uport, can actually, based on that document and based on the ethical approval that we have, get these tissues to the researchers, taking away the threshold that it would normally give them if they wanted to start this on their own. And so we can now do that for many different cancer types, and we've provided thousands of tissues already to all kinds of different research groups.
It's very interesting. It's very reassuring for me to know that these kind of ethical considerations are actively taken into account, and that we've learned from historical mistakes with traditional cell lines where huge things were going wrong. On a different note, so knowing this is a very interesting technology, it can give us very interesting answers to research questions, generate hypotheses for treatment. Okay, we're going back now to the field of peritoneal surface malignancy, and you've already alluded to your work with Organoids on that. Where do you see us in 5 years' time with this? Where do you think that this will lead us? What will be the points of connection with the clinicians? Do you foresee trials based on outcomes of or results of your organoid work in peritoneal surface malignancy?
Yes.
Maybe before embark, I asked the question because one of the things that we've learned as peritoneal surface oncology surgeons is that we're doing a really good job at the surgery. We should be very happy with ourselves at that point. We standardize it. More standardizing is, but as one of our friends told us, eventually this is not going to determine the results. So we've learned that the thing which is not working or not working well enough is the IP chemotherapy, and of course we're looking to people like you to come to us with suggestions. Should it now be monotherapy, or should we apply the medical oncologist view that any good chemotherapy is multi-cycle, multi-drug? Is there like results or pending results or insights from your lab saying that, well, probably it's going to evolve this or this, and we're going to address it in the lab by doing this?
Well, I think this is really, so I think that there are two sort of approaches that we can take to improve treatment. One is indeed use the organoid technology, and we recently built this biobank of peritoneal metastasis-derived organoids. We now have 54 organoids derived from peritoneal metastases, and this we are actively using as a platform, first of all, to indeed show that if you give them oxaliplatin or mitomycin C, heated for a short period of time, from half an hour to 90 minutes, that the vast majority doesn't care, really. There are some organoids that really have a hard time after that, But all of them, all that we tested, will very rapidly grow back. That's for sure. So that's basic. And then we're using concentrations of both drugs that are also in the flush in the HIPEC setting, or even higher. Yeah. So basically, this is never going to work. If you put the drug with the tumor cells and there's nothing else there, and then even then they don't care, then I don't think it's going to work in the patient.
Okay,
So
so from there, that so basically you're confirming from the translational setting that the negative trial indeed was a negative trial and was bound to be a negative trial.
And one could think, what is the rationale for using oxaliplatin as a single drug for the treatment of these very aggressive tumors? You told me that there is, I didn't know it, that there was one trial on oxaliplatin monotherapy given systemically for systemic metastases. But
it has a response rate of 16%, which is like dramatic.
Yes. Okay, but how many people were cured by this treatment?
Yeah.
We're looking at a patient category that is going for intentionally curative treatment in the HIPEC procedure. So we want to kill everything with the current so this is where the organoid technology came in for us with the current two drugs that we use, and we also use irinotecan, which is starting to be used intraperitoneally too. We cannot kill them.
So what should we do?
What should we do? So we published one paper with Inge Ubink a few years ago. One of the things that tumor cells do when they are challenged with mitomycin C, there was a study on mitomycin C, is that they activate their DNA damage response. So ATR gets activated, and that pauses the cell cycle and gives the cell the time to repair its DNA. And then once the challenge is gone, which is, it's only there for a short period of time, once the challenge is gone, they have repaired their DNA, they resume the cell cycle and regrow. And this is exactly what we observe in the organoids. Now, the field of DNA damage repair inhibitors, like inhibitors of the CHEK1 kinase and the ATR kinase, is really, there's a lot of clinical studies with these inhibitors. Multiple pharmaceutical companies have created inhibitors, have generated inhibitors that can target this DNA damage response. The idea being that if you disallow tumor cells to repair their DNA, and if you disallow them to pause their cell cycle, they will accumulate DNA damage and they will continue the cell cycle, and that will in the end be detrimental to the cell. Now, this concept we tested in organoids, and Inge published that, as I said, a few years ago. And now with the new biobank, we generated a far larger data set. And this turns out to be a really, really effective. Heat is not needed. For instance, one thing that we tested, heat is not needed. It works with inhibitors from different pharmaceutical companies. It works with WEE1 inhibitors, with CHEK1 inhibitors, with ATR inhibitors, and in particular in the combination with mitomycin C. So I think if you talk about, generating proof of concept or translating this to the clinic, I would love to do a clinical study, maybe not immediately a HIPEC study where we would need many patients in order to get a survival benefit, because that would then be the endpoint. But maybe first in patients with inoperable metastases, see whether, for instance, in the PPOC setting, where we have repeated access to the abdominal cavity, and we can actually see what's going to happen to these tumors as you treat them with monotherapy or with the double therapy. And this is also how I envision sort of the stepwise development of improving treatment, that we test biological concepts, in this case mitomycin C in combination with DDR inhibitors, and we now test that concept in patients with inoperable disease. But so that means that we have a substrate to look at over time. And in that sense, of course, the PPOC setting is ideal because you go into the abdominal cavity every 6 weeks. Yep. Right? So that gives us
access to new material, to both access pathologically and
and that's actually the second platform that I think is going to be really helpful, this sort of PPAC interact-like studies, having regular or repeated access to the abdominal cavity, because this is one of the very few situations where patients with metastatic cancer can actually be followed in this way. So we can learn much more from what we're doing treatment-wise and how they develop over time than we're currently doing. And you are aware of the first PPAC study that of which Jans de Hing was PI from the Eindhoven Hospital. And actually, we did this in that study, and so we learned a lot from what oxaliplatin does to the peritoneal metastases. And of course, it's not going to or it didn't cure the patients, but we have learned an immense lot of what is happening in the peritoneal metastases when they are given 6 times or 5 times or even 3 times oxaliplatin. And based on that knowledge, so direct analysis, there were no organoids involved there, right? There's just analyzing clinical specimens over time. And in that sense, I think the abdominal cavity is, and the repeated access to it is a gold mine in terms of drug development.
Yeah, two comments to that. First of all, that's really interesting. One of the things that we underestimate as surgeons is that we have the access to the material, to the biopsies, which is like, especially if you do it sequential as in HIPEC or other models, which is like really thing. The second thing is, and this I really you're underestimate a little bit of the work you just shown us. This is for me the first time. So the thing about combining mitomycin C with an inhibitor and showing or demonstrating a clear proof of principle that this works in organoids and the potential step taking this to a clinical model. This is the first time I see a translational research in peritoneal carcinomatosis with a direct impact on a clinical research proposal. So no longer extrapolating from other studies in other metastatic sites, no longer like blindly just trying out a protocol. This is like a research-driven proposal for a clinical trial, of which the impact cannot be underestimated. And I see it with great pleasure, because I think this is the way to go, and the way a lot of this is one example, but there is a lot of research going along similar lines with different models, different settings, but along similar lines, that proof of principle in a model, controlled environment, make a research proposal, and then take it to patients and look which of the settings is now HIPEC or PPAC, or try to once again reprove it before going to a clinical endpoint in a clinical trial, of course.
Yes, I completely agree.
Mm-hmm. I think that more or less for me like concludes, and I'm so happy with this, more or less like concludes the message we wanted to confer to the rest of this audience, that first of all, clinicians and translational scientists no longer are on their islands. We have like a very energizing cycle of new insights coming from different methodologies, and this is one, and all of these methodologies are quite complicated when you first look at them. But even as a clinician, we have to understand that the answers will come from this setting, and that we actively have to open a dialogue with people like you and people on the same lines, trying to build, first of all, good research questions, which it all start. Think actively about the model we're going to use, and this is like a really interesting and really like exciting one, and then build towards a clinical setting. So that is for me like so nice, and once again, I want to thank you for all the great work you have done and are currently doing. And we are absolutely looking forward to your work in peritoneal metastases once this is published. So I happily with you make appointment for a next podcast, preferably in 2-3 months if you can.
Okay, we'll see. But if I can shortly remark on that, we about 12-13 years ago we decided exactly this. You can only make progress, really implement your research, or if you want your research to have clinical impact, the only way is to generate a lab, and in our case that's now called lab translational oncology, where clinicians and people with a biomedical background are working side by side, shoulder to shoulder, work discussions together, retreats together. This is how the lab has been built up over the past 10 years. About 50% of the people in the lab have a clinical background and want to become surgeons, medical oncologists, radiologists, etc. The other half has a research background and they want to become researchers. But they share the lab. They sit next to each other in the same culturing hood. In the work discussions, we're discussing both clinical problems as well as what, for instance, the DNA damage response does, which proteins are involved, etc., etc. And that's so energizing for both for people with both backgrounds, and that, yeah, we found that this works really well. So if people want to set that up, then please make sure that clinicians and biomedical researchers are physically together in the same lab, have the desks next to each other, etc. So that for me, that's I think the basis of all of this.
I couldn't be happier with that kind of hopeful conclusion to our talk, and I want to greatly thank you for sharing your time and your insights with us today. So thank you, Onno.
Oh, my pleasure, Kurt.
See you
It was
soon.
my pleasure. Thank you. See you soon.